a wonderful time at oh man, what is our fifth ending or was our fifth ending age-related disease conference? And I think I'm in agreement with Steve, and I think that was our best one yet, right? So virtual high five there. Don't leave me hanging.
Hygienic high five.
Yeah.
If anybody watches Scrubs.
Yeah.
So yes, it was their fifth, and I agree, it was superb. We are, I believe, back in New York next year.
I hope so, or
So
somewhere live.
But we might be doing, we're entertaining potentially doing a hybrid event. So it's physical and virtual. So that means everybody can get involved. So there we go. Yeah, it was great, wasn't it? We had some interesting stuff. But I think today is particularly interesting, today's topic.
Yeah.
And we've been waiting a long time for this one.
Yeah, and actually, apropos to the meeting, one of the principal investigators on the paper, Irina Conboy, spoke at our meeting, and she talked about, I believe, some of this data and a lot of other data. And this paper just recently came out. So a very good talk from Irina at our conference, and a lot of other great speakers. Steve Horvath was there. George Church was there. I can't remember, but it's a lot of great. Greg Fay was there, on and on and on. So we've got everything posted. So please take a look at all the talks, all the questions, really good stuff. And we'll go live next year, hopefully, with our conference, and maybe we'll switch it up and be it in metaverse at some point. I don't know.
Yeah, it's also worth noting that if you did have a ticket for the conference, you can still log in. And you can still watch all the talks on demand right now, and they should be there for at least a month. So if you didn't catch all the talks, or you want to watch them again, and you got a ticket, you can go and watch them again. And we've already released Keith's opening talk on YouTube, on our YouTube channel, and we are going to be steadily releasing the content in the coming months. So keep an eye on the news outlet. So anyway.
Yes. Anyway, back to where we are, which is our journal club. And as I mentioned, one of our speakers at the conference was Dr. Irina Conboy. So if you want to hear more about this work, please check out her talk that we have available somehow on our website from our conference. So the paper that we're going to be discussing, just introduce myself. I'm Oliver Medvedik. And welcome everybody here to the Lifespan.io conference here, journal club series, not the conference series. That's for next year. So the paper is Old Plasma Dilution Reduces Human Biological Age, a clinical study. So really cool. And what this is all about, this is from Kim et al. And Irene Conboy's and Michael Conboy are, I believe, the principals, final authors on this paper, last authors. This is from UC Berkeley. And basically, what is old plasma dilution? There's a couple of terms for it. Plasma dilution is also referred to as plasmapheresis, and it's also referred to in this paper as TPE, which stands for therapeutic plasma exchange, not telomere position effect. I just kept getting confused about that because I've worked in a yeast genetics lab. So therapeutic plasma exchange. And what is therapeutic plasma exchange? Well, we've heard about over the years blood exchange between old animals and young animals. And people have been doing studies where they've actually exchanged young blood into old subjects and noticed improvements. And of course, there's a lot of issues with that. You have to have whole blood. So you're limited in how much you can obtain. There's also, you have to do a lot of testing of the blood and so on and so forth. And so it's not very feasible, not very readily applicable to wide-scale human use. And the reason people have been doing this is that they've noticed improvements in the older organisms when they've done experiments, which are referred to as parabiosis, where you connect the circulatory systems of two organisms, usually mice, some sort of rodent. But we've noticed improvements, physiologic improvements in humans that have taken, that have done this type of blood transfer. But again, not very practical. Plasmapheresis is a bit different. It's basically blood dilution using, I believe, an isotonic saline solution with albumin supplemented into it. to basically bring it to physiologic isotonic concentrations, right? So you're essentially diluting the old blood rather than adding in new factors from a young organism. So so there's still some, still studies being done to determine whether or not some of the improvements that have been known physiologically have come from young factors or, positive effects of factors that are in young blood, or whether it's in a dilution of negative factors. And I think, don't hold me to this. I think when we spoke with Irina, I think, she stated it could be, there could be a contribution of both. But what this paper shows here is there is definitely an effect, a positive effect with just a dilution of old factors using this technique of plasmapheresis. And another big positive about plasmapheresis, which is also known as therapeutic plasma exchange and plasma dilution, is that it's already been used in the clinic for many decades. So it's not only a simple administration versus having whole blood from a donor, from a young donor, and all the controversies that potentially can ensue and issues. But I believe it's already being used as an for various diseases. I think Guillain-Barré syndrome, various myasthenia gravis. I think in some trials of Alzheimer's. But it's been used for I believe, I think some of these are autoimmune type disorders, and diluting some inflammatory factors is shown to been efficacious. So this has actually been used clinically and been prescribed clinically for other indications, not for aging per se, but for other indications for lots of decades. And I don't know when it was first applied, but I think I may have found a paper going back into the 70s or late 70s or early 80s. So quite a long time and a very good safety profile, right? So this kind of checks off all the boxes. Safe, been in use for a long time. Can we transfer, this type of treatment, this, old plasma dilution, and see into, older cohorts of individuals and see whether or not you get an improvement in a variety of, effects that are known to increase with aging. And the pleasant surprise in this paper is yes. So let's take a look. All right, so this is a summary, basically, of sort of the findings of this paper. You have your older fellow on the left and somebody hopping around with a cane, all happy on the right. So TPE-treated aged blood, healthy. So again, therapeutic plasma exchange. So, essentially, it's a resetting. It's believed that, if you're removing a variety of factors contributing to inflammation, and we'll take a look at some of these panels that they do, by diluting with a dilute plasma solution that's supplemented with albumin, you have a variety of effects that cause a resetting of sort of a lot of protein pathways, a lot of signaling pathways to a more useful state, right? So inflammatory response, DNA damage, and something interesting they call biological noise, which we'll take a look at in a moment, goes down, right? So all these factors basically go down. So let's take a look at some of their evidence here. So they do this with some patients. So they've got a whole bunch of different samples here. Let's take a look at where they mention samples. So they have samples 1, 2, 4, 6, and 7, and 8 from old individuals. So people from there, from 60 to 77. Anybody who's 60 here, I apologize. They call you old. And samples 3 and 4 were middle-aged, 46 to 52. Oh my God, I fall right in between that. All right, so middle-aged. I would consider myself young, but okay, I guess I'm middle-aged. So we have these samples here from individuals, and their treatment paradigm is basically these are the rounds of therapeutic plasma exchange. I don't know off the top of my head how much plasma exchange you need to do. It's not the whole blood. You're obviously not exchanging everything because you need your red blood cells and white blood cells. So I don't know exactly off the top of my head how many liters, whether it's 1 or 2 liters. Probably that's about right, I would think. And you have pre, so R0 is pre, so this is your person before you get an exchange, and then post. And post, I believe, is a month, right? So they do this monthly. So R1 is after month 1, so R0 is pre, and then this is 1 month after plasma exchange, and you do another plasma exchange, and then that's a month later, so 5 months. And I think they followed up these patients for up to a year. And they do a number of different assays. So they take the serum out, they profile the proteome, see how that changes. They look at DNA damage, specifically this oxidized guanosine, 8-OH guanosine. They also look at a senescence marker in the serum, P16. And they also look at the RNA, the transcript. They do a transcriptome profile, basically see how that changes. So, and they look at the immune status of the patients. So this is sort of kind of a one like snapshot here. And just by looking at this, you can see trends, right? So all these colors are different patients over time. The colors basically mark those patients that I mentioned earlier. So sample 1, 2, 3, 4, 5, 6, 7. Yeah, there they go. The different ages you can look up, right? I think green and orange are middle-aged, and they also trend as well. So, instances of DNA damage go down. P16 senescence marker goes down. So month to month, which is, I don't know, it doesn't look like it's even like plateauing yet, right? So there's still some room to go, perhaps. You can keep this up. So one thing that I'm noticing here is that, this is definitely something that, fairly long-term, but fairly innocuous, right? If this is being done once per month. So this is, I don't know if here they have on these samples here, they have a young cohort. So this is S1, S2, 3, 4, 5. So I don't know what the, right off the top of my, they do that for other samples later, but I don't see that here. But the trend is pretty evident in relative expression. So CD3 cells, right? CD4 cells, helper T cells, cytotoxic T cells. Your T cell number goes up over time. Your B cell number goes up. Your natural killer cell number goes up. And there's also a ratio that is skewed in older individuals, lymphoid versus myeloid cells. And that ratio is basically reversed to a younger ratio, which is more indicative of a more resilient, youthful state of the immune system. So they look at white blood cells here. So the trends are pretty impressive, especially considering that this is, a sample size of 8 patients, right? So this looks like a pretty, you don't need to do really any deep dive statistics when you look at kind of, the trend lines like this, when you look at a graph. And I'm sure this would be much more robust if you, had 100 patients or 1,000 patients. So nonetheless, these are pretty steep curves. And again, orange is middle-aged and green is middle-aged. So even those are trending in the right direction. I'm gonna stop sharing there for a moment. So, and I don't think they've done any experiments in this paper to do again, these were kind of long-term, right? So, pushing like 6 months and more. I don't think they did tests of like behavioral tests, right? Like to see if cognition improved or whether or not people were able to run faster or, their gait improved, right? That would be interesting to do, right? Because that's like, you fundamentally want to see if somebody is actually, if these physiologic, changes actually translate to somebody feeling better and acting better and being more mobile, for example. So kind of more observable changes. So that I'm not sure. So that would be, I would say, like the next thing I would, love to be looked at.
Yeah. Oliver, may I jump in?
Yeah, sure, of course. Anybody.
So I don't want to nitpick, but I mean, like I think you also said it. Like you would expect here to see a young person, like a young control and old control. It would really have helped with looking at these graphs. And also, like the normalization that they use in, I think, all of the graphs except for the DNA damage. So they normalize everything according to R0, but I wonder what are the absolute values, like whether we see a bigger change when we start in a worse shape, or I don't know, whatever we would see if it wasn't normalized this way.
Right. So I mean, that kind of both of those critiques kind of boil down into is that a does that translate into a significant improvement, right, for I guess a patient's health, right? Granted, this has been used for decades and has been noted to offer, I don't know at what level, but definitely significant enough that people still do this clinically for various autoimmune disorders. So there's clearly a medical benefit for other disorders. Whether or not this is a significant physiologic improvement for an aged person, trends are there, but is this significant enough? And if not, is this protocol optimized as of yet, right? Because I don't know, maybe you need to do more rounds of plasmapheresis or less. This is done monthly, and I'm not sure. I'm guessing that this protocol is probably adopted from standard plasmapheresis protocols that are already, approved by the FDA and other authorities for other conditions. I don't know if this is for the clinicians to kind of figure out, is this optimized for, the treatment of age, right? Yes.
Can I comment on this? Why I see this as being really significant is because you have this debate as whether there's something in the blood, something in young blood that makes people younger, or something in older people's blood that makes people older. And I think this goes strongly to argue that there is something underpinning the aging process, which is in older people's blood. I think it's interleukin-10 personally, and I have my reasons for that. And I've linked on the chat to some research that indicates that interleukin-10, which is part of SASP, actually causes cells to become senescent at times. So, I think where this is really in the broader aging debate, this particular paper and the general one indicates that there's something in older people's blood that grows, that generates the Gompertz formula. So it's a process which is self-reinforcing, a self-reinforcing feedback system that actually causes cellular damage. I have my own views as to what that cellular damage is. But that's why I see this as being particularly significant. I think this says it's something in the blood.
Well, I mean, I agree. It's clear that there's a component in the blood, and it's clear, I think, from this paper that that there's a negative contribution from elderly blood. That being said, there could be components that are positively acting that are still kind of pieces to the puzzle that we might not get a full reversal of aging effects just from plasmapheresis alone. That's something we need to still tease apart to see how much of a contribution there is from.
I'm not saying that plasmapheresis alone is something that could maintain steady state or reverse the deterioration from age, but I think it would go a long way in that sense.
Yeah.
But what I'm looking at is the theoretical aspect, which is what is underpinning all of this. I don't mind explaining what I think is underpinning all of this. I think you've got this question of programmatic aging, hyperfunction, all sorts of different debates. The difficulty a lot of the sort of methylation arguments encounter is, what if it's something in the blood? Because if it's something in the blood that's affecting the way in which genes are expressed, which is what I think it is, and it's something in elderly people's blood or elderly creatures' blood that creates a greater problem, then that doesn't fit with the sort of programmatic theory.
Mm-hmm. Yeah, my we can certainly debate like the different theoretical aspects of aging, but I'm fundamentally in the entropy camp, and that there's a lot of different ways to, for entropy to be achieved in an organism, and some of them may predominate in other systems, whether it's dysregulation of the epigenome, increased cross-linking, increased misfolded proteins, point mutations in somatic tissues, so on and so forth. Then you have mechanisms in place that seek to export that entropy in a variety of ways, right?
That's fair enough, and we take different views on this. But the challenge for any, you look at the various things that work. Rapamycin, I think, is accepted to have some effects. Exercise, hypoxia, all sorts of things. Whatever system there is has to satisfy all the known facts, and I think this paper gives us one of the known facts. And one of the known facts is that in people's blood as they get older, there is an increasing amount of something. And it may be more than one something. I'm not, much of the time, an interleukin-10 person, and I can explain why, but much of the time, an interleukin-10 person, it could be that there are a number of different things. Although, as somebody with a mathematical physics background, I'm inclined to think the Gompertz formula implies there is one key driver for this, because of the Gompertz formula having essentially that one key factor. I am impressed by this, because I think there was this debate about whether it's something in younger people's blood or something in older people's blood, and I think this resolves it, that it's something in older people's blood.
Mm-hmm. So, any other comments here?
Yeah, if we could just talk about panels D and E for a second.
Yeah, sure.
So this is the part of Figure 1 where they're looking at their blood cell counts, and I'm a little confused by their
panel B or
D as in dog.
D. Okay.
D and E. So I'm a little confused by their methodology here. My understanding is they're doing qRT-PCR to on markers for various blood lineages. Mm-hmm. And I don't really understand why, because most of the time when you're doing quantification of blood cells, you just do a complete blood count. You flow the cells and you just count, you look at surface markers.
Yeah.
So I don't really know why. Do you understand why they did it this way?
No, I mean, I would, like you said, I think when people usually do this for blood, they just put it through, they do a FACS, right? Fluorescence activated cell sorting, and you basically label them and you get a plot, and then you see what the ratio percentage of the cells is. It might just come down to the tools they had at hand. That's kind of the simplest answer, I would think. Yeah.
There's one other aspect that confuses me a little bit. So the big cellular changes in the immune system with aging, it's more myeloid, less lymphatic, right? So mm-hmm. They're looking at, in panel D, they look at all of them, basically every lymphatic lineage there is. I'm a little confused as to, when they look at myeloid, they only, if I'm understanding panel E correctly, they don't look mm-hmm. at it directly, they only look at it in terms of contrasts. So there's no CD69, or there is CD68, mm-hmm. But only versus something else, as opposed to just looking at it directly like they do in panel D. Mm-hmm. And that also kind of just confuses me a little bit, because I guess I just would like to see that data directly.
Yeah, I'm not sure why. Why it's, I mean, I think it may have been referenced in an earlier paper that was the ratios that was observed for elderly subjects. And they just use that as a metric, but I'm not sure if the data is someplace in the supplemental where it's just absolute values. Yeah. So yeah, like you said, seeing populations of cells in the blood, definitely it's more typical, especially in immunology papers. You see a lot of FACS plots of cells being clustered, maybe this was easier for them. They didn't have a fluorescence activated cell sorter with the right labels, and this was just this qRT-PCR was just the fastest way they could do it. That's my guess. We could certainly ask her. Okay, so in the next panel, they actually do have the controls. Here, let me see if there we go. So here they're looking at serum, from a variety of different individuals and doing antibody arrays and basically mapping the different types of proteins they have. And I think, are they doing also expression? No, these are antibody arrays of heat mapping of 72 proteins that are significantly different, almost a twofold change between young and old cohorts, and a principal component analysis to compare the differences. And these are all the factors that kind of change. So here you have young, and I think young is folks that are in their late 20s. It's in the paper, versus old, which is that old cohort group. And then you have patient samples here. I think this would pertain to one. So R5, right, or let's see, R3, R2, round 2, round 1, round 0. I don't know why you have less for round 3, 4, 5 at the end here. But this is basically an expression profile of different proteins that are correlated with, different states of youthfulness and aging. So the 72 that most change, and they characterize them in a variety of different biological processes. Most of them are signal transduction. A lot of them are an inflammatory response. So you've got your whole range, and some are in apoptotic regulation, so on and so forth. But the trend here is that when you look at, old versus young and see how they cluster in this principal component analysis. So old being red, this, circle here, and young being this blue circle here, and then all of your various, folks, middle-aged and old. As this treatment progresses, these arrows are basically pointing, from I believe R0 to R5, which is after 5 months or so. You have basically the protein, content, or the protein levels match closer and closer to that of your young controls. And that applies to proteins that are involved in a pro-inflammatory response. So just looking at the expression levels, and also negative regulation of apoptotic processes. So you have this trend basically going towards the more useful direction. Does anybody want to comment on any details here any favorite protein amongst all of these? You've got toll-like receptors in here, various interleukins. They do mention a big one. They mentioned that a lot of changes have happened. Like there's like one central protein that's kind of like the most, I think, out of all the proteins that are upregulated, it's the most connected with most of them pathway-wise, and that's just jumping ahead. TLR4, which is toll-like receptor 4, which is involved in the innate immune response and binds, I believe, to lipopolysaccharides and a bunch of other bacterial components.
If we're looking for favorite proteins, I'm actually looking for two. So I'm looking for IL-1 beta, and I'm looking for IL-6 because they're some of the most potent, like pro-inflammatory interleukins. Although I'm not sure I see them unless they're named differently.
Could be. There's still IL-1 beta. Nope. At least not on this. This could have just fallen off the chart because they said the 72 most, changed. It could have, that's difference by 1.75 fold. So if that one was at 1.5 fold, it wouldn't have made the cut.
Right.
At least not here that they show. So okay, so you have all these changes occurring. So proteomic profiling, and here they look at yet even more pathways that are basically related to telomere position effect. So various cancer pathways, and this is what I was referring to here: gene expression levels, and that go up most. They do go up for this TLR4, toll-like receptor 4. So when they do these protein-protein interaction maps, they note that TLR4 is kind of the hub that's most connected with all of the pathways, and that does go up with aging. So it is like I mentioned before, it's a receptor involved in the innate immune response. I guess infections go up as we get older. So there's a reason for this particular, I don't know what else it connects to, probably a lot of other things as well. But that is one of the key receptors that goes up. And a lot of these pathways that are, for older individuals, you get an enrichment, obviously, in these pathways that are involved in cancer development, a lot of signaling pathways. And these pathways are, again, the trend is towards a young expression profile after multiple rounds of therapeutic plasma exchange. Keep wanting to say telomere position effect. God. R0, R1, R2, R3, R4, R5. So old. So again, they've got the controls here, old and young. And yeah, if anybody has a favorite signaling pathway they'd like to talk about.
I have a comment. I'm not sure. It's really weird. It seems like this figure 3B and figure 2B are like the same big heat maps. They're just the same heat map. They just changed the order and they also rotated it. So I think it's the same thing.
Same thing, but basically they clustered the path. Well, actually, you know what? JAK-STAT signaling pathway. Key pathways related. You might be right about that. Is it the annotated, rotated version of the top one? CRLF2.
I looked a bit and tried to contradict this hypothesis, to reject it, and I didn't manage to. I looked at some examples. And also here, the other heat maps here, E and F here, I think they are also just, they took some of the rows from figure 2B. A bit weird. You have one heat map and three other panels that are just the same data.
Could be. It could be. Here they basically, maybe they just wanted to cluster this as in a form of signaling pathways. And because here the only difference is that obviously they will annotate, and they said, well, these are involved in or upregulated in pancreatic cancers, right? So SMAD4. So that gives you this extra information here. So is that the same data? Maybe I'd have to take a. I don't think I have the time right now to zoom in, but we've got the paper here. I don't know if that's the same one that's basically copy pasted and annotated on the left. It could be the same data.
Not the same order, so they changed the order because of the clustering. But yeah, just wanted to mention it. It was a bit weird to me.
Okay, yeah, agree. It is more useful seeing it this way, though. If you've got the pathways annotated here and clustered, that makes more sense to human eyes, right? So rather than having something like this, which is also useful because you see the trend, but this is sort of an alphabetical list, and this neatly puts it into categories that you can see which of these signaling pathways, which clusters of proteins are involved in which signaling pathways. Actually, it's a little different here. Some of these are repeated, right? Because there's going to be overlap, like TGF beta signaling pathway. You have TGF, I just noted here, TGF BR1 and TGF BR1 are here as well, and here. So it's a little. So I think they just reordered it, and you do have in this row here you have repeated ones. So those are all distinct up there, and here they're just rearranged and clustered into these signaling pathways. So I think it's a rearrangement so to better illustrate how these cluster into different pathways, if that makes sense. So yeah, I just quickly zoomed in to see if there's like IL-2RA is in JAK-STAT signaling, and IL-2RA is also in PI3 kinase AKT signaling as well. So, which would make sense because there's a lot of overlap with signaling pathways. Okay. Good eye there, Oren.
Can I talk about my favorite pathways?
Oh yeah, sure.
Well, the Janus kinase is interesting, but I sort of look at things in a broader sense, which is a question of what genes are expressed. And that's where the Janus kinase comes in, because obviously that controls NF-kappa B, and as a consequence, through the citrate carrier, the expression of genes through acetyl-CoA in the nucleus. And my view is actually that's actually what we should be looking at. Why are cells not producing all the proteins they should produce? Why are all the genes not being expressed? And it happens to be it goes through the Janus kinase, the JAK-STAT route, that actually a lot of that's affected. But I think you need to look, rather than looking at like the consequence of the genes not being expressed, you need to look at the question that the upstream effect, which is that the genes are not expressed. And if let's look at it. We'll go back to the 1948 papers looking at the epigenetic changes for the queen bee, and that the queen bee, because the queen bee has royal jelly, has different genetic expression compared to other bees who have exactly the same gender, same sex rather, and the same genes. And here we have this question: as people get older, they express fewer genes, their cells don't function as well. Why is that? I happen to take the view that it's through the Janus kinase, yes. But I think that's the thing to be trying to, if you're looking at the broader question of, and this is where the relevance, this is such an important paper, because it says that there's something in elderly blood that grows that prevents certain genes from being expressed. I think that's what it says. But if we forget about the individual items, there are lots of individual items in it. The broader question is what's actually going on. And obviously I have my own views, but I think it comes down to this broader question of why are fewer genes being expressed for older people or older animals? And I, as I said, I think it's to do with IL-10.
All right, I will definitely look up more on IL-10. See, we have
I'm doing the IL-10 stuff. IL-10 is part of SAS, all right?
Mm-hmm.
So as a cell goes senescent, it produces a number of things. Now, IL-10 is interesting because it's actually strictly anti-inflammation. And what it does, it when it hits the Janus kinase in a cell, it down-regulates NF-kappa B. And when you down-regulate kappa B, NF-kappa B, you reduce SLC25A1. That reduces, as a consequence, the amount of acetyl-CoA. So it reduces the acetylation of the histone, not the methylation, but the acetylation of the histone. And as you reduce the acetylation of the histone, then you express fewer genes, so you don't produce as many proteins. And then I would hypothesize that makes a cell senescent. And there was a good paper in 2021 looking at the question of osteoporosis, which said, okay, osteoporosis results from a shortage of acetyl-CoA in the nucleus, which comes from a lower expression in stem cells of the citrate carrier, SLC25A1. And there you have what I like about that as a hypothesis is it's got a feedback system. So as you increase the amount, and actually when you look at the Portuguese paper I linked to, which is not published, it's a bit sad that what I think may be one of the most significant papers ever happens to be somebody's research for their PhD that was never published in a journal. But I've linked to it in the chat.
Yeah. So it depends on which sites we're talking about when it comes to histone deacetylation, because there's a lot of work obviously with sirtuins where an increase in histone deacetylation. So deacetylation going up, it relates to an increase in lifespan and an increase in genomic stability, right?
Well, this is where these are really interesting questions, and it comes down to what is it that happens to older cells? Why are older what function? If we look at satellite cells in muscle cells, for instance, okay, they don't. When people get older, sarcopenia results from them not producing the right sorts of protein. Why? When you look at all the things that happen as people get older, the cells don't do everything they used to do. And the evolution has produced these genes that produce lots of different proteins. But actually, as people get older, they don't. Why? Right? And, the fact that there isn't the acetyl-CoA to acetylation. Acetylation and deacetylation, it's a dynamic situation. There's acetylation going on, deacetylation going on all the time. And it is really quite complicated. But you have this question, why? And then if we're trying to actually say, what is it that happens to people as they get older that stops their cells functioning as well? Why? And I think this paper is brilliant because it says it's not something in younger blood, it's something in older blood. So we need to look at something in older blood. My view, it should be something in SASP. All right, because obviously senescent cells develop as people get older. If we're looking for a feedback system, as there are more senescent cells, so you have to look at SASP. And I picked the IL-10 simply because of the 285 or whatever number they are, I think 785 inhibitors of NF-kappa B, which affects the citrate carrier. That particular one has that feedback system, and in fact, that paper, that research done in Portugal, which was never properly published, as not published in a journal, rather sadly. There's lots of other papers that substantiate that as a thesis. But so I like this for that, and I like to look at the broader questions, because aging is a broader question. It's not a narrow question. It's like, why are we not mole rats? Things like that. But what is it? What's the broader thing that causes most animals to age and not be long-lived turtles? And I agree. This is why this paper, you had obviously the heterochronic parabiosis, which is ethically something that causes all sorts of questions, and is not really practical in the long term. But this says, well, it's not actually the need for young blood; it's actually the need to not have old blood. That's what it says. I put aside all the minutiae of everything. It's coming down to that very simple point. There is something in the blood.
Yep. And speaking of broad strokes as with regards to aging, I want to get to figure 5 because that's actually, I find that one actually quite interesting. I've never seen this sort of correlation done before, and there may have been some other papers. So I'd like to hear people's input on this. But getting back to figure 4 and the changes that happen with components in young versus old. They seem to home in on this other component here, TDP43, which I'm actually not too familiar with. It's increased in the blood of patients with ALS, Parkinson's, and various dementias. It's a regulatory protein. I forget what it stands for. I think it regulates, involved in transcriptional regulation. Is anybody familiar with this protein that's kind of upregulated in various dimensions, TDP43? I Googled it. I took a look at it. I think it's one of these proteins like a TAR protein, which is basically regulates transcription at the level of the mRNA transcript, kind of similar to what happens with like HIV transcripts. But I'm not too familiar with it. But regardless, this correlates with levels of this correlate with various neurological disorders that they've noted here. I don't know how else they came across this. So they did, just reading this paper here. So circulating biomarker of neurodegeneration. And they're looking at this one specifically. And the levels go down of this marker. Relative TDP43 levels. Let's see, how did they measure that? I believe that's these transcript levels. I think they're transcript levels. Doesn't say here. So other than that, I'm not an expert on TDP43, but it seems to be a pretty broad neurodegenerative marker. I don't think I've actually introduced a paper in any of our journal clubs on TDP43 up to this point. neurofibrillary tangles, amyloid beta plaques, but no TDP-43. It's another protein I need to look up. So the levels go down. So if anybody has any comments on this as a biomarker for neurological disorders, again, I'd like to see this translated to physiologic responses, right? Because if these levels go down, are these people actually getting cognitively improved? Is there a cognitive enhancement?
So, sorry, Oliver, I think I'm missing something. Why did they single out this particular protein?
Yeah, that's what I'm looking at here. It's obvious that the levels are higher in young versus old. I don't know, out of all the different kind of markers for neurological disorders. I'm just looking at the paper right now. Age and neurological disease-specific clustering of systemic proteomes and the reduction of a circulating by TDP by TPAs. Let's see. They looked at a data set here. They did a uniform manifold approximation projection analysis of defined distinct clustering of old people with Alzheimer's disease and AD-related diseases, so Alzheimer's disease-related diseases. Considering these observations, we decided to analyze systemic levels of TDP-43, which is trigger of several neurological pathologies. Good question. I don't know why specifically TDP-43.
Maybe you just had a really big fold change on their array.
Yeah, probably. They just jump in the text too, considering that, obviously a whole bunch of markers related to neurological disorders go up with age, but why specifically it was TDP-43. Actually, this is not transcript. This is TDP-43 specific enzyme link. So this is an ELISA assay looking at TDP-43 levels, I believe, in the serum. Okay. So yeah.
The first panel is unrelated to TDP. I think it's more general, right?
It's more general. Yes, it's more general.
And actually, about panel A, I'm not clear about which patient exactly they chose for this and why.
Well, yeah, it's well, I think they have only one patient, if I'm not mistaken, right?
No, we have 1, 2, 3, 4, 5, 6. I mean, we have 7 or 8.
Okay, so A is age dementia, distinct UAP clustering of 72 proteins that differ between young and old for 6 age and disease groups. This is sample 1, 2, 3, 4, 5, 6, 7.
No, the colors are different in panel A. You can see the panel A has a different legend.
Oh, right, neurological disease. Sorry.
So it's like
yeah.
The red and purple are only the ones from this experiment.
Right.
The TPE.
Yeah, you're right. I got confused by the color transition here. Yeah, I don't know what the patients are. Unless they say it in materials and methods. It might be in the supplemental. I don't see it mentioned here. It's definitely not in the figure. And I don't see it in the text. Let's see. Comparative bonus compared to healthy age matched in young cohorts. Figure 4A in supplementary table 1. So it's a data set that they got from somewhere. I don't have the supplementary figure for that data set right now. Let's see if I can pull that up. If there is a link to the data set here. usually on the bottom of the paper. Okay, well, if anybody can pull up the supplemental, that would be great.
Which figure do you need?
Figure 4A. I believe there's a supplemental 4A.
Yeah,
and
I have it. It's an Excel table, but it's just in general the whole population. Like I can share if you want, but it just says the ages and the gender of people and sex of people
okay.
in the study.
Right. So each of those points is a patient sample, right?
No, I don't think so.
You don't think so? Okay.
They're too close to each other. They don't say anything specifically with regard to this figure. So I guess they just chose one patient, because otherwise the patients wouldn't be so close to each other. They are different ages. Some are middle-aged, some are old. I mean, it couldn't be that they're so similar.
Yeah, I don't know.
If I had to be pessimistic, I would say it was just one patient that fit the conclusion that we want to see, right? But that's the pessimistic side.
For what? For figure 4A or for figure B and C?
Yeah, for 4A.
4A for one patient for
the TPE. Like they don't say how they chose it.
You mean one patient for each of those clusters?
No, I mean we have the TPE R0 and the TPE R last. The question is which patient is this patient?
Oh, you're talking about just specifically TPE R0 and TPE R last. Okay.
The others are like control.
Gotcha.
Not control.
Yeah. Yeah, I would assume that TPE R0 and TPE R last would be, well, I would assume that it would be multiple patients, right? Rather than just one. But I don't know that off the top, and that's not reflected in that Excel spreadsheet.
It couldn't. I mean, it sounds impossible that these are multiple patients because they are not the same age. We saw some of them are middle-aged, some are old, and obviously they are different. Everyone is different. Humans are this annoying thing. I don't know.
Okay. Good question. I don't know. We'd have to. Yeah. We probably have to ask the authors that if we don't have that in the supplemental. It's not something that any of us can answer probably at the moment. Okay, so moving on to figure 5. This one is kind of interesting for me because I've never seen kind of data. I don't know, Michael, any of you guys have seen like this type of analysis done, which is a increase of variance or standard deviation being used as I guess I'll call it a metric of entropy, right? They call it biological noise. And they don't look at like, I think they look at the ones that change the most. And they mentioned somewhere that not all of them do this, but enough of them do. And I'm not familiar with the technique they use to pull the ones that they're looking at right now. They refer to it as, just going through my pages here. One second. What's the technique that they used? Too much terminology for me to learn. I underlined it. Okay, never mind. I'll get back to it. I can't. Oh, the Benjamini-Hochberg procedure. Yeah. I don't think I've ever applied that procedure in any data sets. So what they're seeing here is for a number of proteins. And I think one of them that they look at has the most change, which is this plasminogen activating receptor, right? It's urokinase plasminogen activating receptor that's involved in immune responses. They're basically looking at expression profiles, I believe. These might be transcript levels directly, I think. So transcript levels, I think. And what they're looking at here is basically the standard deviations, right, of transcripts in, I guess, clusters of individuals, and that these standard deviation values, right, for at least these sets of genes, these expression profiles.
It's protein levels.
Protein level. Oh, is it protein
It says
levels?
protein levels, yeah.
Oh, okay, sorry. Standard deviation. Oh, yeah, right. Yeah. Of 507 proteins, antibody array proteomics. You're right. Sorry about that. So protein level standard deviations go down in an aging transition. So young to old transition, and also the yellow is pre to post therapeutic plasma exchange transition. They go up in old to disease transition. I believe the disease here is neurologic disorders, and middle age to old transition to a slightly less extent. But sort of like and the mean standard deviation values go up, and then when normalized to young groups as well. They also refer to this as biological noise. So here we have individuals in their 20s, 30s, 70s, I believe. It's E70s, L70s.
Maybe it's early and late 70s.
Perhaps, yeah, because I think they have people from 70 to 78. And then pre-TPE to last round of TPE. So they basically convert this into the standard deviations as a histogram to biological noise for a number of these proteins. And then they try to, convert this into kind of a clock, if you will, sort of like a Horvath clock, but in this case, a noise clock that corresponds to chronological age, and making this into sort of a biological age metric. And I found this intriguing because, sorry about that. I found this intriguing because I don't think I've ever kind of seen that as a demonstration of age as a measure of stochasticity using this type of system. I think they sent papers that were fairly recent, 2 or 3 papers. And I don't know, Michael, have you seen something like that?
I'm actually on a preprint that's looks at this sort of stuff. Although it's with RNA data, not proteomics data. Who did they cite? Like Amir Bashan is another guy who's like done transcriptional coordination with aging, which is like not exactly the same, but it's kind of similar.
Yeah, I think they cite Nikopoulou from 2019, which is, if you have the paper, it's citation 62, and citation 63, O'Laughlin from 2020, and maybe Nordstoke, which is from way back in 2007, but that might just be a statistical technique they use. I think 62 and 63 that were like the 2 references they cited, and those are like, again, pretty recent references. So
I'll look those up. Maybe I should be citing them.
Yeah,
I might be able to comment a little bit on this. Like, I'm not familiar with their exact methodology. Like the high level view of why this is a cool approach is that lots of stuff increases in variance with age.
Mm-hmm.
And there's really obvious stuff, like some people age better than others. I think everyone kind of knows that. You can quantify that in terms of like, as your grip strength, how your spirometry change with age. And there's lots of variance at the macro level. Some people age better than others. And it looks like it holds true at like the omics level. If you look at gene expression of genes that correlate with age, or where the expression level correlates with age, aside from there being some genes that go up or down with age, a trend which holds true quite often is the variance of expression often goes up with age. And it's kind of like the micro analog to what we already know is true at the macro scale. So when I look at this figure in the Conboy paper, I wonder if that's what they're sort of going for. Like, you might expect that across their sample, maybe their TPE treatment would cause standard deviation to go down across their markers. That would be consistent with the story I told about variance increasing with age. It's kind of tricky though, because like one of the big gotchas here is that stuff where there's a really low baseline of expression has much more variance typically. Like if you have a protein where there's almost none of it, like maybe you have 0, 1, or 2 units, and you're comparing it with another protein where there's like 1,000 or 1,100 or 900 units. The stuff which has a very low baseline tends to get disproportionately represented, and it's kind of one of the big gotchas here. So I kind of, when I see their like what in panel A, B, and C, they've got I guess 6 different. So they got 18 different proteins we're looking at. One of my first questions is like, which of these have higher expression levels and are probably there's less. If you'll forgive that, there's less. Which of these has least noise in the noise measurement?
Yeah, I've got a, I'm really intrigued by this, and I'd like to see like other correlations done, which would be really interesting. Like, well, number one, is this like is this kind of variance apply like everything across the board, right? If we just say like, okay, let's just look at like the entire like all genes that are being expressed, right? How much, is do we in general get a much wider variance or for everything, right? They do mention that there's actually a reduction in variance in a couple, and I think they gave some response. It's somewhat buried in the paper, so it wasn't like an increase in variance for everything, right? So it'd be interesting to see, is it disproportionately more variance? The other question is that does this variance correlate with a loss of regulation in like other things that might be upstream, like methylation status, right? So do you have like, is this is where you're getting most of this disruption or most of this variance happening? Are these areas where, you have the most kind of like changes in the methylation status of individuals as they get older? Because I remember asking Steve Horvath what he thought about the changes in the methylation status, whether or not it was some of these changes were programmed, or did he think that it was kind of a stochastic change in the methylation profile? And he was leaning towards that it was a stochastic dysregulation of methylation status. That was kind of the key driver. That was his I think view on it currently. I'm not going to hold him to that. I'm sure if other data comes in, he'll obviously change his mind, but he currently thinks that's what's driving the change in methylation status. And I wonder if this variance is also reflected in other things like the methylation status, like you mentioned, Michael, with other things like grip strength and whatnot. So I'm just intrigued, right? That if this noise, right? I mean, it's referred to as noise, right? So that is noise is entropy, right? So this is a kind of another measure of a increase in entropy of the system. I haven't seen this kind of type of figure before, so I find it intriguing.
Yeah, I think this comes back to the essential question of what do you measure? There are so many different things that can be measured, and I've done a quick search. I don't think they measure C-reactive protein, which is a useful proxy for IL-6 because it's relatively easy to measure. It's not so easy to measure, but it's relatively easy to measure. And so you get this difficult question of like how it comes back to how do you measure aging, and they have their approaches and various other people have their approaches. Mm-hmm. I think my view about the paper is in the broadest sense it's measuring aging, and in the broadest sense it's saying TPE has an effect, and the problem is when you look on a narrow basis and you look at the question of senescent cells. To me, senescent cells are a key part of aging, and they're patchy. So you get patches of senescent cells in different parts of the body. So you can have bits that are, and I, as I said, I have my view as to why that is, but they are clearly patchy. And so any one particular body will have different patches which are more senescent and less senescent, and that's quite difficult to compare on a quantitative basis. Although, as I said, CRP is useful because it follows IL-6, and IL-6 is not just generated by SASP; it's generated by other things as well. So it's not a perfect measure. But I come back to what do you measure to work out whether you're actually affecting the core aspect of aging, or whether you, in fact, all you're doing is ameliorating some of the symptoms, which is what people normally do.
Yeah, I mean, I guess it all, we're all looking for kind of proxies, right, for the most fundamental metric, which is basically, a decrease in mortality, right, or decrease in the rate of mortality over time, which is, takes time to measure, right? So obviously, if somebody lives to 200, that's going to answer a lot of questions, and you can measure that. But in the meantime, yeah, we're all after these proxy measures that basically can point us in that direction.
And in a sense, Gompert's measure of mortality is quite a good way of doing it, but you're looking for more than that, which is quality of life, health span type issues. So, which is sort of perhaps a step a little bit away from mortality per se, although clearly linked. But yeah, this is the difficulty. There are so many things that can be measured. What do you measure? What can you carry forward from one paper to another? And I must admit, I like their work. I do really like their work. And I take the broader question, which is, without being too picky about any particular analysis, is TPE something that has some merit? And I think the answer is yes. And what does it say? It says there's something in elderly blood that grows that we should be looking for. And that's, I think, the essence of the paper. When you look at papers, I think it's quite good to sort of, not necessarily to look at the minutiae of it, but to say, what in the broadest sense is that telling us? And I think that's what it tells us.
It could be useful to measure things like hand grip strength, how long you can stand on one foot, and particularly sarcopenia and cognition.
I believe John just froze right now.
No, John Ferber froze. I didn't freeze. John's still frozen.
Yeah.
I think he's right.
John Ferber, not John Hemming. Yes.
Yeah, I know. I'm not frozen. I'm just gently getting.
He'll come back.
John was into cryonics, guys.
He'll come back.
So he's definitely gone into. He's done a good job, and I think he'll come back to life soon as well.
He's successfully in cryonic sleep now.
Yes, I think he is, but I do think he's going to survive. I think he's going to be a good example of somebody surviving. But he's right about sarcopenia. Sarcopenia is a good thing. How you measure it is quite difficult. But I think sarcopenia. I've been doing my own experiments and measuring. Oh no, he's gone now completely, hasn't he?
Yeah, hopefully he'll come back. But yeah, I think so. I mean, this TPE has been done for many decades now, and probably. The kind of shame is that there's probably a lot of data that's lost, right? Because you do these treatments on people, and nobody's measuring all of these other parameters that relate to aging, right? It's being used for a bunch of other disease applications. So I mean, this is a wonderful opportunity. I think I may have mentioned this at an earlier journal club, or maybe I even mentioned it to Irina. I'm not sure, but I think it was actually during a journal club with you, Steve, where I'm, you could certainly set up, sort of a trial protocol where these metrics are measured by people that are already undergoing these types of treatments, and see whether or not there's like improvements in a variety of parameters that relate to aging, right? So a lot of different metrics that could be basically measured. Blood samples are obviously taken, right? So you can certainly analyze them for a different type of profile that reflects changes that you would expect, in the aging process, as well as carbonite process as well. It's back. So I think, there's a lot of opportunity here to capture that data for people undergoing therapeutic plasma exchange. And there's no technical limitation right now. It's more of a procedural limitation, like a trial limitation, right? I mean, I don't organize these things, but something that's been done efficaciously for decades like this, where people already have blood draws taken, is just a matter of setting up kind of a scaffolding, if you will, to capture that data that's being kind of lost.
Yeah, can I make a point about blood draws though, which
Yeah.
is I with all my own personal biohacking stuff, I most of the time do weekly blood tests. I stopped over the summer because I went to Spain, and through that process, I've learned which of the biomarkers are reliable and which ones are not reliable. So the fact that somebody has a blood draw that's been sitting around for a time doesn't give you necessarily that much good information, because there are very clearly biomarkers that change as the blood. If you freeze it sufficiently, you're all right. That in itself causes problems. So, this is the problem. You can measure so many things, absolutely loads of different things. And one of the things I regret myself with my own experimentation is not measuring enough right to start out with. And the more you measure, the more you can potentially find out. And what you need, and this comes back to the biological age question of, how do you measure progress? And as I said, I think CRP is a really good thing because it's not perfect. It can change from exercise and all sorts of things. But on the other hand, it gives you something reasonably comparable that's not too expensive to measure. And sadly, they haven't measured it.
Yeah, that's something worth asking Irina here, right? Because these experiments with the TPE have been going on for for at least about a year. So it would seem to be pretty easy to measure various other physiologic parameters, right? Like lung capacity. Gait,
all sorts. But there's
like
so
that.
many things.
Yeah.
I've got the devices to measure grip strength, and grip strengths are useful. But the problem with grip strength, you see, is it's like muscles, right? Developing muscles is three factors. One is the protein input. Another one is exercise. And another one is the ability of satellite cells to differentiate. And when you get those three right, you don't get sarcopenia. Particularly sarcopenia, I think, is the differentiation problem. But measuring it, how do you measure it? It's very difficult.
Yeah, I got a quick question here that's very basic, and maybe should have asked Irina this during her conference, but I just was thinking about other things and blanked out. Sometimes the most basic questions kind of elude you. Maybe some of you know this. Where do they get the albumin for this? Is it recombinant human albumin? I mean, obviously it has to be young albumin. It can't be old albumin. They can't put the old albumin, or are they putting old albumin back to make the isotonic solution, right? Because it's an isotonic saline solution. And the only protein I believe in the therapeutic plasma exchange is albumin, if I'm not mistaken, or at least the only one mentioned. I'm assuming it's not an animal albumin, like from eggs, because of risk of allergic responses. It's recombinant human albumin.
I don't know the answer to that, but I know that albumin is a medical volume expander, like that's something you use a lot of blood loss.
Yeah.
Because there's so much albumin, I think they're doing 5% solutions, and because the volume's so high, I don't think it's recombinant to be really expensive. My guess is it's purified out from blood products, but I don't know. I don't know how else you do it.
Unless it's bovine albumin.
But then, like Oliver said, you're going to have an immune reaction.
Yeah, although I don't think they tend to use bovine stuff that much anymore. It used to be when they pioneered it, like in the First World War, they were using it.
Sven, you got a question or an answer?
Well, a bit of an answer at least. I also don't know for sure, but my speculation is indeed that it's just purified from human blood. And I also don't think it matters if it comes from old or young people because it's a single protein. The difference with the therapeutic plasma exchange is that you remove bad proteins from old people, but that's, albumin is albumin. Of course, old people's albumin may be a little bit more damaged, but of course, as you know, it's a turnover, right? So the protein doesn't last that long. So I don't think that even if you were to compare the amount of glycation, for example, on albumin from old people versus young, I don't think it's going to be that different. Of course, with diabetics, that might be different, but for the rest, I think it's not really that different. And so it's just probably taken from blood from people.
If I may, that actually might not be true. Thinking about it, so albumin, one of its major roles in physiology is it's a carrier for hydrophobic stuff. Like albumin is a big carrier for steroids, and it's a big carrier for fatty acids. If you're doing cell culture, like you can get different grades of albumin, like Albumax has a really high lipid content. I don't know how albumin is sourced or purified, but there could be differences in what your albumin's binding. Like you could have hormone high albumin, or you could have like the stuff that's used in cell culture sometimes, fatty acid high albumin. And it's not an easy thing to like standardize because albumin tightly binds like a lot of stuff. So yeah, now I really am wondering how they source this.
So it's kind of the two. The simplest answer would be that if they basically take your blood out and basically, I mean, they could isolate your albumin, right, and just put it back minus everything else, right? So like maybe I don't, but they would have to purify it, so it would probably not be like very practical. It would have to be sourced somewhere else. But like, there's like somebody's tap dancing in the background somewhere. Okay, so yeah. Yeah, I don't know if there's if there's a paper out there where people have measured old versus young albumin. Like, that's a good question. Like, is old albumin like not just let's say there's nothing. Yeah, let's say maybe there's nothing wrong with the albumin per se, but maybe there's like other stuff that's glommed onto the albumin, right? That's going along for the ride. That's like a negative factor, and maybe you can get the TPE to be better by using younger albumin or albumin that doesn't have. I don't know. I'm just thinking out loud here, tossing these ideas out. How is albumin sourced? Probably pooled from a whole bunch of different, donors.
Well, that would also be an interesting question, because if you pool it from 1,000 people, then you will get some type of averaging of whatever is bound to it.
Yeah.
Which is probably a little bit my guess, if you want to, because as Michael, I think it was, who previously mentioned, it is used in medicine, so it is actually consumed in relatively big quantities. This is not something that is used in milligram quantities. This is like, every day in hospitals used in, I don't know, kilogram, whatever quantities. So it probably is produced in very high amounts. So then I can just imagine an industrial process where plasma from thousands of people is mixed in one giant, container, one giant incubator or something, where it's then being extracted.
I'm surprised they don't have massive amounts made for safety and purity's sake, like recombinant human albumin, like they do with insulin, just like throw a whole bunch and, I mean, if it's such a huge product, right? You would think that, you would have fermentation tanks making recombinant albumin. I don't know.
The problem is it's just expensive. Like if you got a fermentation tank and you can make 10 grams of insulin or 10 grams of albumin, like you need nanogram quantities of insulin and you need gram quantities of albumin. It's just a much harder thing to make cost effective. At least if you're going for equal standards of purity.
Yeah. I have a humanized horse. Celiac. Why not, right? I mean, it's just a technical challenge. Humanized horse, humanized immune system with, for a horse. And that's
another.
Maybe that's a market opportunity. Maybe that's a market opportunity right there.
Another interesting question then is, how is the risk of using this? Because if it is purified from humans, there is always a certain risk on transmission of HIV, hepatitis, and so on. So, I mean, of course, if you want to do this from healthy aging perspective on a regular basis, you want to be absolutely certain that this is pure and clean from any possible risks.
Right. I mean, if you are, yeah, I mean, if you like when people undergo dialysis, right? I mean, I guess I could do some homework here and look this up, right? Like you basically put the albumin back to somebody if you undergo dialysis. You end up taking out, urea and a whole bunch of other metabolites. So is the same thing done in plasmapheresis? Do they just recycle the albumin back into an individual? You spin out the is there any way you can actually have a column that can take out the albumin? And I don't know. I don't know. So obviously you do column purification. So the technical answer is yes. The practical answer is I'm not sure if there's a practical, dialysis methodology for that to be achievable at scale. That's the real question. Maybe it is. I'll take a look. I'll do some searching, and next time we meet, I will hopefully have the answer.
What I would say is that if producing albumin was the requirement for people to have a quality health span, then I'm sure people would manage to find a way of doing it in a more cost-effective manner.
Yeah. Well, it's certainly there. There is already a methodology to do that right now in plasmapheresis and has been for many decades. So I'll take a look at what the standard methodology is just to enlighten myself and see if there's any improvements on that methodology. But anyway, it's a very promising approach. Definitely a much simpler approach than whole blood transfer, less risky as well for obvious reasons. And should be much more cost effective. And the other check mark here is, again, this is an approach that's been used therapeutically. It's been approved by the FDA already for a number of different therapeutic applications that are not aging per se, but for various immunologic disorders. So this should be a pretty low hurdle to kind of overcome to get into human trials to see if the physiology of an older person actually improves using this technique. And yeah, that's kind of my take home on this. I think it's very promising, and I'd love to see more follow-up papers where they look at much more many other much more parameters that are related to aging, kind of at the physiologic level, lung capacity, gait, cognition, and see if there's improvements in that as well. Versus cellular biomarkers, because certainly we're seeing an improvement in that. So I'd love to see if how much of a translation there is. Be interesting to make a correlation between how much of a change you can get in cellular biomarkers and how much of a difference in improvement that can actually correlate to at the physiologic level, right? So yeah. That's kind of like my 2 cents here, right? We have these biomarkers as a proxy for aging, but I wonder if there's a kind of a much more fine-tuned correlation we can make with change between certain protein expression levels, so transcriptome levels, methylation levels, and another proxy of aging, which is physiologic response, right? There's a twofold increase in a certain interleukin translate to a twofold increase in grip strength. I'm just like totally, obviously not going to be that kind of, probably dramatic correlation, but could we do a proxy to proxy correlation from cellular to physiologic, right? And I looked at kind of that'd be cool as well. I'm not sure if anybody's doing that. I
think it's important actually. You talk about grip strength, yes, and grip strength is a key thing, but that also depends on exercise, because if you exercise your grip, you get stronger, and it also depends on protein inputs, as with all sort of strength things. So I think the difficulty is you need to sort of look at correlations between steps in the process. It's the same as things like when people talk about
rate of change, basically. Yeah,
it's a rate of change issue. Yeah. So it's really hard to get any useful information when you're initial input is too remote from the output. So yeah, I don't deny that grip strength is a useful measure.
Yeah, I was just using it as an example. It doesn't have to be
Yes, but it's an example. But it's again, it's sort of, you need to try and analyze it down a little bit. And as I said, this thing about senescent cells being patchy, so you start having problems in your kidneys and it gets really serious. The patchiness of senescent cells makes it very hard to get an aggregate measure.
Mm-hmm. All right. So
actually,
yeah,
I have a question about the protein, like the biological noise. How exactly do they measure the biological noise for a single person? I mean, do they take multiple samples? What are they measuring exactly? Because I think for populations, so they test the variation between different individuals, but what do you do for a single individual? What do you measure?
Probably, well, you'd have to look at it, chronologically over a period of time, a longitudinal study, right? I mean, either that, or you would look at averages between populations at a given moment. I don't think there's any other. Those are probably the only two ways you can do it, I think, right?
Yeah. So here we don't know what they did because we have in panel J. I just don't know. That's why I asked. Like,
yeah.
What exactly did they measure in panel J? Is every dot here one of the patients, or?
It would have to be, right? I don't think this. I mean, otherwise, the trial can't be going on for 40 years, right? So this is 40 to 80. So it would have to be different patients.
Yeah, it also matches the I think the ages, because the 77 was the oldest one.
Yeah.
It's also TP. So yeah, what do they calculate for a single patient? What are we actually? How would they measure this?
Yeah. I'm just kind of looking at this. TP reduces biological age. Well, I mean, this is relative years. This has to be. This data has to be imported from the noise measurements they made before, right? So that's where they're getting. I mean, that's chronological age is obvious, but the relative years is probably calculated from the noise. And I'm not sure if.
It doesn't look to me to be very reliable figures, that particular panel J.
Yeah. I mean, relative years, 250 years. I mean, it's, I'm not sure exactly how,
My
is coming from.
Guess for their, if you want their methodology, they look at the whole study population to get standard deviations at each of their proteins. And then, so you have a standard deviation, you have 507 standard deviation values. They then score, so they make their clock, and they probably score individual patients by looking at like what the z-score is for every individual patient across all the proteins. So if your expression value is very different from like the average, that, which is to say, if on a given protein you have a high standard deviation, a high z-score, then that probably that's how the age clock is probably computed. I don't see how else they could have done it.
I do think that trying to claim that people, it seems to argue that people who are in their sort of 80s and 90s changed to being in their 20s, and I'm much that I do think it's achieved something. I don't achieve that.
No, I meant exactly what Michael was trying to answer. Like, what exactly did they measure? Because we have only one person with 77. So what you're saying, Michael, is that like we are comparing to the other people, but they are of different ages. Well, some of them are middle-aged. So it would be weird if this is right, or I didn't understand what you're saying.
It's a bit of a reach. If they use the method I described.
Yeah. Also, another question. I mean, if I just take protein levels and then I dilute the plasma, so now the standard deviation should be lower because I have less protein of everything, right? So if I just take a random variable and I don't know, divide it by 10, then the standard deviation would also be divided by 10. So I wonder how do they correct for this? I mean,
well, things would have to be the same concentration, right? I would guess.
what do you mean same concentration?
Well, you're asking how is your question, how do they correct for different dilutions of different samples? Or
yeah, I mean, in general, the concentration is lower by definition, right? You dilute the blood. So let's say you dilute it 5-fold, right? You dilute it. So now you have 0 proteins except for albumin, right, in the blood. It's not zero. It's not zero, right? Because
when you say dilution, are you talking about dilution in the patient's body? Like
TPE. I mean, the TPE itself, the procedure, is lowering the concentration of protein in plasma, right?
Yeah. I think practically speaking, like the percentage of actual blood that's done, I don't know off the top of my head. I don't think it's like how many liters you have in your body versus how much is actually converted using TPE. I think it's not a very high percentage. Otherwise, you're going to jeopardize. You're going to dilute out hormones and all sorts of things. I think that's it's not. I don't know what the percentage is. It's something I could look up. But I don't think it's like a really, really dramatic increase, right? I don't think it might be 2 or 3 or 4-fold higher than how much is removed when you donate blood. But it's, even when you donate plasma, it's like 2 liters, right? Versus how many liters are in the human body? It's like 40 liters more.
4.5.
Just 4.5 liters?
Yeah, 8 pints, 4.5 liters.
Google says 5. Yeah.
5. Okay, 5 liters. Okay,
I think it's 4.5.
Okay. How many? So you donate actually pints. So how many pints is that?
8 pints.
8 pints. Okay, 8. Okay, so 1/8. So
I don't know what they did in the thing, but it's the other thing you have. Obviously, you dilute the blood day 1, and then over a period of time, the cells generate various bits and bobs.
Yeah, 40 liters would be a huge person. It's a whale.
I just forget the 40 liters number. Let's go for 4 liters.
4 liters, okay.
4.5 is the figure I tend to rely on. Google may say 5. I think 4.5 is probably.
I'm obviously a cell biologist. I'm like, how much blood is in a person? I'm obviously a cell biologist. How much blood is in a person?
Yeah, I know. It's one of the. I'm a bit of a generalist. I'm actually a physicist. But the
somewhere between a gallon and 1,000 gallons, somewhere in between there. Oh.
Yeah, well, one way or another, it's sort of like
You're trying to fill up a swimming pool or something, Oliver.
Yeah.
I mean, 40 liters. I don't even think an elephant's got that much.
Yeah, well, I don't think it'd fill a swimming pool either, but that's another matter.
40 liters. I mean,
anyway, whichever way it is, the blood, the body will generate some more stuff and shove it in the blood.
Yeah.
So and obviously the blood's in a dynamic situation with things coming in and out of the cells all the time. So
well, when they do the blood draw and actually do the analysis, I think they mentioned that it's 1 month after they do the R0, which is then they do the plasmapheresis, and I think they do the analysis like at the end of the month before they do the next draw.
Probably sensible because it gives you some stability. The difficulty of these things are all dynamic. And this is what one of the difficulties is it's all dynamic.
Yeah. So your blood, I do know. Yeah, your red blood cells are turned over in about 3 days. So I think
120 days for red blood cells, please.
120 days. Okay, I'm off by the number.
That's like HbA1c is like 3 months or something like that.
HbA1c is a different beastie anyway, because that's 100. What you have is you generate new red blood cells and they die off over a period of time, and it's roughly 120 days cycle time.
So we can calculate how much percentage is recouped by the end of the 4 weeks, right? And figure out. I mean, so
Yeah, but this is serum anyway. This is not the red blood cells.
Yeah. But I mean, as far as what Oren was asking about how much dilution there actually is, like month to month, right?
Yeah, the serum. This is the difficulty, because obviously blood is including the red blood cells as well as the serum. So I don't know. Do we have a figure as to how much they're actually diluting yet? Do we have that from the paper?
It's probably
It's in the methods how much they replaced. Was it 5% or something?
5% of serum or 5% of blood?
Ooh.
There you go.
Have a look at the methods.
I'm trying to figure that out now. It's not clear. I think the solution that's intravenously added is 5% albumin by weight. Yeah. Then how much of that is
what
there?
What volume is it?
Well, that's going to be in references 20 and 21. So TP was performed as described in references 20.
They don't answer it either. It's
a Scooby-Doo mystery, guys.
Oh, there we go. Yeah. What do we got? 2021.
And I would have got away with it if it wasn't for you damn meddling hallmarks.
There's a newer version of this article available, it says.
See, we can Google it. How much actually in a plasmapheresis clinic is actually removed, right? I mean, that's a standard amount, I'm sure, like for these protocols.
So the best answer I can give you is that the AMBAR study, which does plasmapheresis for Alzheimer's, does like 20 grams of albumin. That's not exchange because they're not getting rid of stuff, but it's like the closest equivalent, because that's also an aging associated study, and it's still adding albumin. Actually, I'm sorry. AMBAR did 20 grams and 40 grams, so those are their two levels.
But that's albumin itself rather than albumin in saline solution, or what?
They did albumin in saline solution. In fact, I can link you the company that sells their albumin, but you'd have to back calculate the volume.
If it's 5% roughly, say it's 5% by volume, then let's just assume it's like same mass as water.
Right.
20 grams?
20 grams, yeah.
20 grams. So at 400?
It's 400, yeah. But
400 milliliters, it's not really much. It's so we need to. To me, it doesn't affect the paper, because basically the paper says what it does, which is that it's in the old blood, and you have a broader question of how to deal with that particular reinforcing feedback system, which is, yes, this may be something that's a useful thing to go through, and for me, and I think I may be the chronologically oldest person on this call at the moment, and so it may be something I should consider trying out with all the various other things I try out over time. But actually, what this paper helps us do is get to the bottom of what the reinforcing aging, the self-reinforcing feedback system is behind aging. That's what I think is important about it. Not necessarily how much albumin is injected into a body with so much saline or whatever.
I mean, John, I think it's really important to make sure we got right the small details before we.
The small details are important, but I still don't think whether it's a liter or half a liter or 400 milliliters, I don't think makes any difference.
If everything is accurate and everything is perfect, yes. But we also have to think about that when we read the paper. We want to be critical and.
I do like to know these things, but I don't think it makes a substantive difference to the underlying issue.
All right. Have we wrung all the blood out of this and serum out of this paper that we can possibly?
That's terrible. That's terrible. Oliver, you've been told about telling jokes.
All right. Well,
you know, the police are going to get involved if you carry on.
The dad joke police?
I got an answer for you guys. Where does the albumin come from? So for Ambar, that company, Grifols, whatever it is.
Yeah.
Their product information is that it is a cold alcohol fractionation from pooled human plasma from venous blood. So that probably means they get expired venous blood that doesn't go into transfusions. They get it from a lot of people. It expires, and they biochemically fractionate out the albumin. That's probably how they get so much without it like breaking the bank. So yeah, the big question: what's the age of the donors?
Maybe old albumin is good for you. Maybe.
Or average albumin is good for you.
Yeah.
Maybe young albumin is better. Who knows?
I'd rather have a younger vintage if I can, really. It's not like fine wine, so.
Yeah, wouldn't that be something, right? It's not that old factors are being diluted. It's old albumin. That's the trick.
Nah.
I know, but
old albumin is structurally not as good as fresh albumin. It's just not. Which is something that Irina was talking about. So there you go.
Well, I'm wondering now if another control can be done, an experiment where we can have different types of albumin.
Yeah. We could have an alcohol control group as well. You know, dilute your blood with lots of alcohol. Does that make you younger?
I'm up for that experiment.
I'm definitely up for it, but I'm going to be honest, I don't think it does make you younger.
But what I would say is take Pantherine. That's something I've learned in my 60s, and I would say Pantherine is very helpful for alcohol.
So yeah, unfortunately, a nice bottle of port isn't gonna reverse aging, but it might make you feel a little bit better about it. Wouldn't it, Oliver?
Yeah.
Or whatever you do. You go to McSorley's, isn't it?
I don't really go there that often. They have only 2 types of beer, light and dark, in their own brand. By the way, that is a reference to the oldest operational bar in New York City. Not old by UK standards. I think 1859. So older than the Civil War in the United States, but definitely a young pub compared to the UK. But the oldest, definitely old by US standards, and it's right across the street from the Cooper Union. So if any of you guys are in town, I'd be happy to show you around and take you to McSorley's, and you can dilute some of your blood plasma with beer.
Yeah.
Meanwhile, in the UK, I can literally walk around the corner and look at a building that's older than the entire country that Oliver's sat in. Just saying. But yeah, if you want to go to a really old pub, I'll take you to the Trip to Jerusalem, which is a pub built into the walls of Nottingham Castle, and it still serves beer. So yeah, it would have been around when Robin Hood was around. Well, hoods. There were many of them.
All right.
Anyway, so I think you've drained the plasma from this one, as you were saying.
Yeah. So I'm definitely looking forward to more plasmapheresis papers coming up on the horizon. It's definitely something that people are already doing. So let's get the data. Let's not lose it. People are participating in these trials, and is it affecting aging at the physiologic level? I'm optimistic. I think all the arrows seem to point to a positive direction. So let's get that data and see if that holds up.
Well, thank you for everybody for participating in this.
Thank you.
I think we've got to the point where we should probably move on.
Yeah.
All right.
I am. So we'll be.
I've still got some whiskey to drink, so I'll be all right. Don't worry.
Hey. I've got a coffee. There's a bit of an wherever you are. I think we're what 6 hours here. I don't know where you are.
Yeah, no, it's I'm in Birmingham, which
okay.
is Birmingham, England, not Nottingham. And it's 10 to 7 p.m.
Okay, so I'm going to switch to that later. Right now, I'm in coffee stage.
Well, you're welcome to notionally join me in a notional whiskey now.
Caffeine.
Just pretend you're in the UK, Oliver, where the rule is if it's 10 o'clock.
Caffeine to nicotine transition. Caffeine to alcohol transition.
Although I've seen people drink pints of Guinness at 6 a.m. in the airport, I kid you not. So but 10 a.m. onwards is usually it's okay to drink time in the UK. So just an FYI.
All right. Okay, folks.
I do go to the pub for breakfast, but I don't drink alcohol in the morning.
There you go.
All right.
And on that alcohol related note, we should
not good for longevity, I'll tell you that.
It isn't, and the data really actually does support it.
It supports the argument it's not good for longevity, yes.
Oh yes, there was one recently. We should probably do that, Oliver, actually do a deep dive into booze and see whether it is or isn't good for longevity. There is a debate that some is cardioprotective, but I think it's a little bit highly debatable. So we should dive into that one of the days and see what we can conclude.
Well, certainly lower concentrations definitely work for happiness. So, quality of life does go up higher, and quality of life is important.
It does.
It's the HPA axis you need to think about, yes.
Yeah. Okay.
Right. So we will see everybody probably about the same time next month.
Sounds good to
the
me.
Last Tuesday. And what was it you were thinking of doing? Because you had another paper in mind, Oliver. You mentioned.
Oh yeah, I don't know if maybe we'll revisit something else. We were looking at the paper that basically had the microneedle application of two drugs. I think one of them was something from a green tea extract, and I think the other one may have been metformin. This was for hair.
It was rapamycin, wasn't it?
Rapamycin, yeah.
And the green tea,
yes.
The green, rapamycin, which is. I find rapamycin interesting, actually.
Yeah,
so restored hair color, I believe. We should definitely cover that one next week, and I'd like your professional opinion on that one.
Okay.
Because I've got quite a bit of silver hair, so
yeah, I think it was also regrowth. I don't think it was just hair color, but yeah.
I don't need hair growth. I've got very thick hair, but I've got a lot of silver, so if we can get rid of the whole silver surfer look.
Yeah, the other papers that I was looking at were basically papers related to the project that I'm working on with Keith and others affiliated with Lifespan, which is basically non-pharmacological interventions that may ameliorate or cure Alzheimer's disease.
Is this the Leeway Psi?
No. Well, it could be there, but there's a lot of other, Anyway, don't want to go down this rabbit hole right now, but there's lots of papers out there that we can definitely look at from.
I'd go for the rapamycin one. I think a good look at the way rapamycin works.
Mm-hmm.
We like
green tea extract. Yeah, no, well, I've got some, but I haven't tried it yet. But I'm doing well with the hair regrowth and everything else like that. So without trying rapamycin, so I've got quite a happy bunny really. But I do intend trying it later this year, but I've got to get it into the test cycle.
Okay, so rapamycin we might have rapamycin.
It might not be, but we'll see. We'll let you know through the usual channels.
Yeah, it's an interesting one.
I think I'm just going to go out on a limb here and say that I think rapamycin is one of the most promising existing drug candidates. There you go. There, I said it. But we'll maybe talk about it next time.
Yeah. Okay. So see you all sooner than we're already at the end of this month, so we'll see you at the end of next month.
Sounds good.
We will.
Thank you, everyone.
Thanks for everybody who's joined us, and thanks for everybody out there in Facebook land as well.
All right.
We'll catch you next time.